Okay, welcome everybody. I'm Lee Ellis, Vice Chair for Translational Medicine. Is it possible to turn the lights down just a little bit, if you would, please? I want to welcome you to Plenary I, which is focused on translational medicine. I'm not going to go through the outline of speakers today because you can read this yourself, but I think we have a very interactive program, and I like getting the participation of the invited speakers in determining the titles of their talks, and even with CMAC, how to help clarify that for the group. I do want to take one moment to thank a few people. I want to thank James Ray, who couldn't be here, for leading most of the reviews for translational medicine that comes through SWOG. So JR, I hope you're drinking a beer right now up in Michigan with your friends and family, and thank you for your hard work. I also want to take a moment, we haven't done this before, to thank all the people behind the scenes here. As you walk in, you pass by the SWOG desk and you pass by the Hope Foundation desk. And I think it's important to recognize that we exist because of the expertise of all those people out there, the perseverance of those folks out there, and their commitment to helping patients with cancer. And we do that through your efforts along the way. So please take a moment to thank the folks outside who do the hard work for SWOG and for the Hope Foundation. And I will tell folks that there are going to be lots of slides, including some that I showed today, where you may want to take some pictures. So have your phone ready. And if you want to take some pictures, at least of my slides, that's perfectly fine, because I put these up there to advertise the opportunities from the Hope Foundation and some other things as well. So thank you all. And these are the presenter disclosures. I'm tired of seeing people put this up for 1 millisecond and then move on. So you have a chance to actually read through this. And more disclosures. I'll give this 8.9 seconds. Okay, I think that was fair. So here's where you may want to pull out your phones. There are some opportunities from the Hope Foundation. This is their 30th anniversary of supporting SWOG. Please take a look at the deadlines that are coming up. We do have this in the proper order, and great opportunities, especially for young investigators, or better to say early stage investigators. I should take back the word young. And also here you have a chance to be able to access some of the stories that shaped Hope, the voices and some interviews, and Hope's timeline. So take a picture of this if you'd like, and then you can access this information. And Hope exists because of all of us who donate for a good cause, and please consider that as you recognize all the great work, some of which we'll see today, which has been supported by Hope, the career development, and the food that you're eating right now, is powered by the Hope Foundation. So a couple of updates here. I do want to update after our meeting yesterday with the biobank team headed by Nilsa Ramirez. Here you can see that we have specimens in the bank which are ready to be accessed and investigated so that we can learn more about the biology of cancer, the biology of resistance to therapies, etc. So it's a shame to have all these specimens that are well annotated sitting in the bank, rather than people using these to investigate the biology of cancer and the biology of resistance to our therapies. So please take a dive into this and see if there are specimens on Navigator that could help you in your research. And I want to end my introductory comments with the idea of possibly having a special omics seminar, as we had at the meeting in San Francisco. We had a genomics symposium. 6 companies got up there, talked for 10 minutes, and then exhibited the rest of the time, so you could pick and choose your omics platforms. And then I have a question, because San Francisco, there are lots of companies that are just down the road. If you would like to have a proteomics symposium, like a Thursday morning that would last just a few hours, or a ctDNA symposium, I would say put your hands up, but it's pretty hard for me to count from up here, and we don't have that much time. So think about it, catch me in the hallway, and let me know if you're interested in having a symposium on proteomics or ctDNA. With that, I'd like to introduce the first speaker. We'll move on to the general session. Jans Ruder is from the Jackson Laboratory. And normally we have speakers who are initially, the first speaker is supported by the Hope Foundation. The second speaker would be a TM chair or a TM expert from a particular committee. And the third speaker or part of the session is up in the air, and it changes at every meeting. So for the first speaker, Jans, we'd love to hear what you have to say.
Thank you, Lee, for this very kind introduction, and thanks for inviting me. Thank you to the Hope Foundation for inviting me to give this talk. The talk title actually is a question, and that really addresses this very important question in my mind. Can we increase evidence-based genome-informed therapy by implementing a genomic tumor board in the community setting? It's a big question. Spoiler alert, I will not have an answer for you in the next 10 minutes, but I just wanted to start with that. As Lee mentioned, my name is Jans Ruder. I am at the Jackson Laboratory. I'm actually an outlier at the Jackson Laboratory because I'm actually hired as a clinician to run a cancer genomics program in Maine, which I'll briefly talk about. And I also have the immense pleasure of co-chairing S2108CD, which we'll also discuss further in this talk. So when we think about precision oncology and targeted therapies, it's, I think, important to start with some level setting. And this is a graph that really shows the immense progress and the immense work that has been done over the last couple of years to identify more targeted therapies, bring more targeted therapies to FDA approval. And since this graph goes into the future, you can also see that it's very likely that we will see a further increase in targeted therapies, especially in the tumor agnostic setting, which has become the newest field, of course, that really moves beyond just singular biomarkers in non-small cell lung cancer and breast cancer and others. Now, what this means, however, for any clinician is that there is now a significant proportion of cancer patients out there that may have actually a potentially targetable genomic mutation or alteration for which there is an FDA-approved drug that is either available on-label or off-label. And this is a very simple and very rough calculation using about 45 drugs that are listed in this 2022 publication, which was the ASCO Provisional Opinion on Genomic Tumor Testing. There's a very nice table in that paper of the currently FDA-approved drugs. If you take those targets and those tumor types and look at the Gini database, it looks like about 40 to 60%, as mentioned here, of cancers may actually have a potentially targetable mutation. It also leaves, as can be seen in gray, of course, a significant number of tumors that don't have a currently targetable alteration, but a result of that may still be reported on the ever-increasing number of different genomic test reports that a clinician faces. So when clinicians deal with this, they will identify potential treatment options that are currently considered routine clinical care, but they will also have to consider off-label drug use and clinical trials. And then the last piece here, of course, is many alterations that we can now identify as routine testing really are still in the discovery research space. And needless to say, all this information has led to very complex, to a lot of complexity in clinical decision-making when treating a cancer patient on a daily basis. So the question has come up in the community, how can we actually address these complexities and how can we help clinicians to overcome some of the obstacles that they face in this decision-making? And genomic tumor boards or molecular tumor boards are one way that we can really turn genomic complexity of a cancer case into practical options for patients. And I've listed sort of the components that really most, if not all, molecular tumor boards have right there. And we'll come back to this a little bit later in the presentation in some more detail. But you see, it's a very different process in a the typical multidisciplinary tumor board where you're considering the different treatment modalities. Everything is focused around the genomic targets and potential treatment options. Many molecular or genomic tumor boards have been implemented around the world, actually. Most of them are single institution efforts. There are a number of publications out there now that actually show that receiving a genome-informed therapy that's directed by a molecular tumor board is actually potentially associated with better survival. And I just briefly want to highlight the program that we started in Maine in 2016, for which I actually was recruited to the Jackson Laboratory, which is the Maine Cancer Genomics Initiative. This is Maine, as some of you may know, is a very rural state. There's no quaternary medical center, no university-based medical center in the state. And back in 2016, the implementation of genomic medicine and genomic testing was not yet where it should be. So the Jackson Laboratory secured philanthropic funding to start a program that provided testing for free of charge. And we also started a program that included genomic tumor boards and an educational program for clinicians to really further help the clinicians in identifying the best possible treatment options for their patients. We recently published a paper in JCO Precision Oncology describing how we went about doing so, and we're currently working on identifying and analyzing the patient outcomes that are related to this. What we do already know is that at baseline, this is now going back to 2016, 2017, that the level of confidence that physicians had either in their ability to interpret genomic test results or to prescribe targeted therapies to their patients, or for their patients to actually access those targeted therapies was quite variable, and for many of the clinicians actually was quite low. For all the clinicians in the room, including myself, it's probably not too surprising to see that clinicians were more convinced of their own confidence than in the confidence or than in the ability that their patient might be able to access these therapies. Back in 2018, I then had the opportunity to come here to SWOG and actually present some of this work at the Cancer Care Delivery Committee, because it is truly a cancer care delivery initiative that we took. And this led to a collaboration with Meghna Trivedi and others, of course, but Meghna Trivedi and I are co-chairs of this study, S2108CD, which is really the first cluster randomized trial investigating the impact of what we call educationally enhanced GTBs on patient outcomes. And you can see the study schema to the left is a cluster randomized study that randomizes at the recruitment center level. We wrote the study out. It took, as it typically does, a few years before it actually was activated. We activated the study in August of 2022. And you can see a map there of all the sites that are participating. So the entire US is essentially represented with a heavy focus on, all these sites are NCORP sites with a heavy focus on minority, underserved, and rural areas. So in the genomic tumor board intervention itself, this is kind of stuck here.
We advance the slide, please.
Yeah, there you go. I'm going to have to click through this. So this is the same figure that I showed beforehand. We have an enrolling, the physician that enrolled the patient or someone from the study team presents the case. We have a knowledge base at the Jackson Laboratory called the clinical knowledge base. We have a representative from that team go through the variants that are presented on the report and pull some information on clinical trials. We then have a genetic counselor that supports and answers any questions that might come in the somatic report about germline impact of the report. And then possibly most importantly, we have a team of precision oncology experts. These are both PharmDs and members of the Moffitt Precision Oncology team that actually provide a summary of all the available treatment options. All of these sessions are chaired by either Meghna or myself. There we go. And most importantly, actually, as many of you may appreciate, we have a dedicated coordinator at JAX that actually wrangles everyone together and makes sure that these meetings actually happen with all the appropriate information. Now, there is associated education as well, and this is in two different formats. One, we have online modules that are available for all the participating clinicians on the intervention arm. And then we also have additional sort of more informal, if you will, educational snippets essentially that are disseminated via email. So we publish these monthly GTB summaries, and we focus on specific findings of one of the other GTB session that happened. And this is put together by the clinical education team at JAX, and you can see an example of it right here. The study, as I mentioned, was activated late August of 2022. It's going great. I learned this morning it's one of the best accruing cancer care delivery studies apparently ever, which was a surprise to me, but it's great to hear. You can see we're right on target at the moment, and we'll happily report outcomes hopefully in the next few years. So real quick, I have 32 seconds left. So the conclusions of this talk: we can find an increasing number of targetable mutations in most cancers at this point. Physicians still to this day have a varying level of confidence in using NGS testing in practice. Genomic or molecular tumor boards are an important decision support tool for clinicians. S2108CD is the first clinical trial to systematically investigate the impact of an educationally enhanced genomic tumor board on evidence-based treatment outcomes. And I think from this, we and the community, the entire community, really learn what works in genomic tumor boards, how they're impacting care, and which elements we can carry forward. And on second 0, just a quick slide with the acknowledgements. The entire S2108CD study team at JAX, of course, critical for this. And then the funding sources are on the right. So with that, I'll close, and I think we have some time for questions. Thank you.
Great. Thank you. As close to time as you could possibly be. That was great. And you even threw in the line that you were close to time. Thank you. Questions?
The breakdown in terms of disease types, presumably the usual actors, or are you getting rare tumors?
So the question around the disease types. So in neither in the program in Maine nor in the SWOG study do we dictate disease types. It's essentially any solid tumor patient can enroll. We haven't done a formal analysis yet of the SWOG study. I will say there's a fairly significant representation of the usual actors, i.e. non-small cell lung cancers are a significant portion of the patient. But we see essentially it's a good spread, I would say, across the board, including some of the rare tumor types, sarcomas, et cetera. So it's a fairly good spread. In Maine, we actually saw a very significant proportion of primary brain tumors because the clinicians, a neuro-oncologist was very engaged with our program, and she basically sequenced every one of her patients. So,
very interesting talk. I'm just kind of interested, since you're in a rural setting and you're doing all this sophisticated testing, and then you come up with targeted therapies. Do you have then trouble having the insurance companies approve this? Because I've heard from some colleagues who I trained with who went to private practice and maybe in a rural that, hey, you're living in a dream world, buddy, because when the rubber hits the road, you spend hours and hours on the phone, and then, so the testing is great, the science is great, but so now you're in a rural setting where you don't have even an academic center to lean on. And I was just kind of wondering what the follow through is. Do you get the drug that may indeed have much better efficacy, much less toxicity because of the precision medicine approach?
That's a great question. So let me just start by answering this in one way, which is that for every genomic tumor board that we run currently within the SWOG study, but also in the main project, we actually provide what we call curated minutes. So we provide the clinician with a summary of the discussion, including references and the level of evidence that this reference represents. And I'm only telling you this in this context because our thought is that if a clinician wants to appeal a denial of a targeted therapy, for example, they can use that to support the appeal that they may be putting in. So that's one thing that we're doing that's fairly, it's actually quite a bit of work to put this together, but it's not something where we need to change the entire infrastructure, right? Because access to clinical trials would always be the preferred option, but it's rarely, or it's oftentimes not feasible as well. In terms of how many patients actually then started target therapy, we did a preliminary analysis of our data, and it looks like about 15% of patients in the entire cohort actually went on a genome-matched therapy. It's always a question of how you define that. And if you look more closely, there are a number of patients there where you're actually quite surprised because it is truly a fairly unknown target and a targeted therapy that actually got approved as outside of a clinical trial. So it's still probably less than it should be. I completely agree with that, but I think we're making some progress.
Thank you.
Hi, Banu Symington.
Hey, Banu.
I think this is very important work, but with this audience, I also want to say, I think we should all try to hold the companies that do the testing responsible for giving us clinically relevant reports. That's a pet peeve of mine. If we had reports as pretty as the ones that are presented at the tumor boards for S2108, we might not need the tumor boards.
Thank you. So it's a very good point. I think it's a big task to take on, of course, but it is quite baffling that for the same type of information, you have about at least 4 to 5 different widely used templates. And I completely agree with you that that is certainly an issue because you don't have enough time to really go through all the details.
Great comment. Thank you. Great talk.
Great.
Right on time. Thank you.
Thank you.
And we'll move on to the next talk. I think that was a perfect way to start the afternoon. Young, you're on your way up here. Young Chais from Northwestern University has played a large role in the DART trial, and he will talk about TM efforts of the DART trial, building the rare tumor TCGA. All yours.
Thank you. Thank you for the nice introduction. It's a great honor and pleasure for me to be here to tell you about the translational medicine effort of the DART trial, building the rare tumor TCGA. These are my disclosures. All right, so why rare tumors? So as a group, they're really not rare anymore. A quarter of all cancer diagnosed are rare tumors. And as an individual trial, there's not much interest. So it becomes a perfect disease entity to pursue from NCI or SWOG perspective. And there is no known perfect genomic or immune predictor for the dual immunotherapy. So it makes sense to do not a molecular or immune biomarker-based, but a histology-based basket trial. So we do want to replicate the success stories from melanoma, renal cell, Merkel cell, Hodgkin's, you name it. So DART stands for dual anti-CTLA-4 and anti-PD-1 blockade in rare tumors. And for PD-1 blockade, we use Nivo, and CTLA-4, we use IPI. So we're using the low-dose IPI regimen here. Ipilimumab 1 mg per kg IV every 6 weeks, and Nivo fixed dose 240 milligrams every 2 weeks until progression of disease or unacceptable toxicity. And our study statistician is Megan, who helped us tremendously. And we have 3 study chairs for this trial, myself and Drs. Patel and Dr. Kerjrock. So when we approached NCI, we were forced to get married with NCI match, because they were also interested in rare tumors and they were screening all solid tumors at that time. So in order to enroll into DART, they had to be either initial screen fail or subsequent screen fail after they're offered NCI match targeted therapy on trial. So up to 25% were actually rare tumors who got enrolled through NCI match, and this is the NCI match treatment algorithm. But if they meet the definition of rare tumor and screen fail on molecular screen, they can go into DART. So March 2015, I approached Dr. Kerj, I was the chair of our committee, and discussed the idea of pan-tumor immunotherapy trial focusing on rare tumor. And she said there's another junior faculty who has similar ideas, and why don't we all work together? So 3 of us started the DART team, and we were thrilled when this got, the protocol got approved by Central IRB a year and a half later. And in January 2017, the study was activated through the NCTN network. We started with 33 cohort, and it was a very flexible study. So the ECOG group can also, other study groups can suggest the new cohort, and we were able to embrace new ideas. And now 9 months later, because of a lot of things happening with the NCI match, we actually divorced with NCI match, and we were on our own. And then we got the serial blood marker testing started, and to our surprise, we were granted the first CMAC pilot grant, which enabled all these amazing TM effort collaboration. So starting from 2019, we were presenting our cohort data, starting with the high-grade neuroendocrine cohort. And this year, March, we closed our trial for new enrollment. So this was open for a little more than 6 years, and we were able to accrue 798 patients in over 1,000 different site locations. So this was a tremendous success for SWOG, for NCI, and for patient, most of all. So this is the initial 33 DART cohort. And I think what's innovative is that we were able to assign tumor of unknown primary as a cohort, as well as a not otherwise categorized NOC cohort. And this cohort, we were reassigning patients into new cohort on regular basis, which later panned into many different cohorts. As you can see, we ended up having 53 different cohorts, and you can see there's PIComa, there's atheneoneuroblastoma, which I didn't know it existed. And we were able to really just enroll a lot of rare tumor trials and really show the world that yes, we can accrue to these rare tumor trials. And Dr. Kurdrock suggested also biomarker-driven cohort, which is the PD-L1 amplified cohort, which was assessed by the copy number from NGS. So if you go to the SWOGstat.org, you can look at the name of the cohorts and how many numbers patients got enrolled, and these are the sites that had this DART trial opened, including Alaska, Hawaii, and Guam. So I want to emphasize we actually saved lives through DART. In Chicago Magazine, they wrote a story about a patient in early 30s with metastatic adrenocortical carcinoma who exhausted all metastatic treatment options and received DART and went into complete remission until this day, and leads normal daily life. And this is a patient of mine with my whole team celebrating her milestone of disease-free CR with complete remission. She's not on therapy anymore. She had the high-grade neuroendocrine tumor of esophagus. It's pretty rare, but still enjoying remission, and we gave her award, against all odds award. Also, as I was making the slide, I got a letter from a patient who had clear cell carcinoma of ovary, who said she had complete response and is not on any therapy anymore. And she was just interested to know what our patient like her with the same diagnosis doing with the same treatment regimen. So she was just curious and sent me email, finding my email through the homepage. So I thought that was interesting. So this is the select cohort that showed pretty good response rates across different cohorts. And I listed who had how many CRs and PRs. And in high-grade neuroendocrine, you have objective response rate as high as 44%. And you have sarcomatoid lung, you have thyroid cancer, metaplastic breast cancer, small bowel. And you can see that there's gestational trophoblastic neoplasia with response rate of 75, 1 CR, 2 PR out of 4 patients. It's truly rare disease, so it's hard to accrue, but still, we are learning as we go. And we learned that it's really hard to just assume some are cold tumors while there's not going to be any response. No. We even in tumors like granulosa cell tumor of ovary, there seems to be a lot of deep and durable responses. So what's amazing is that most responses were lasting more than 2 years, couple years, and many are still ongoing responses, as you can see in the swimmer's plot here. So the neuroendocrine tumor, especially the high-grade data, led to incorporation of the DART regimen in the high-grade, well-differentiated neuroendocrine tumors, as well as extrapulmonary small cell and large cell neuroendocrine tumors. Also, the data generated from angiosarcoma, as you can see here, led to incorporation of DART regimen in the soft tissue angiosarcoma regimen. Here are the GYN tumor cohorts and vulvar hypercalcemic small cell, which is a truly rare disease. Clear cell ovarian I mentioned, and granular cell tumor. You can see they all produced somewhat deep and durable responses. Some are ongoing until this day. And this is a metaplastic breast data where they had deep response in 3 patients, and all of them still having ongoing responses until this day. And again, this is a collaboration with ECOG. And we have bronchoalveolar data, thyroid cancer, and non-adenoid cystic salivary gland data that showed a lot of PRs with ongoing responses. So what can we learn from DART? I'm going to emphasize that translational medicine is a vital piece of our work. And I do believe that it is the patient's legacy, and it is our duty to pursue what we can, learn from the data, and that make sure that their death or whatever suffering they go through is not in vain. So what are we trying to know? It's really the neglected space, rare tumors, and we have a lot of unknowns. Before the therapy, we really don't understand their genomic or immune landscape, and then to correlate that with response, as well as the immune-related toxicities is what we want to do. So we're building on the data of NCI match because 1/3 of our patients were initially enrolled into NCI match. So we do have those data in handy. And we do require mandatory pre-treatment fresh biopsy or archived tissue if we have it in within 3 months. And then we have 3 different time points at baseline, first imaging at progression-free, at progression blood samples. So with that, we have 2 commercial laboratories as well as CMAC as our main collaborator. Here, we're utilizing the CMAC and CIDC laboratories because we're SWOG. We are working with MD Anderson Laboratories here. And within DART, we are using the tier 1 assay listed here. We haven't gone to tier 2 or 3 yet. So this is our CMAC, CIDC, SWOG team member. We're working on our TM projects as we speak. So we are exploring first cohort 23, 52, which are the high-grade neuroendocrine tumor cohorts, and found and through the tissue analysis, we're sequencing whole exome as well as RNA-seq, and then we are doing the PD-L1 IHC as well as the multiplex IF. From blood, we're doing Olink, looking at soluble proteins, and we're doing CyTOF as well as the TCR sequencing. So among many different findings, I just want to highlight within the neuroendocrine cohort, we found that PD-L1 expression did not correlate with response. However, we found correlation in B cell and dendritic cell signature in tumor tissue with response. And also tertiary lymphoid structure signature was associated with response in this cohort. Immune cell infiltration did not cluster by grade or response. Responses were both seen in TIL present or absent tissues. Olink showed that combination therapy promoted immune mobilization, and CyTOF analysis showed that discrete EMT cell subsets differentiate response at baseline as well as early on treatment. So moving on to the thyroid cancer data, which we just finished analyzing, it showed that PD-L1 nor immune cell infiltration did not correlate with response. However, we saw a correlation with TIL, which were dual positive by PD-1 and PD-L1, which showed nice correlation with the PFS and OS graph. TMB unfortunately did not correlate with response. So what's the next step? We really want to build rare cancer TCGA. The data I show you coming from select cohorts with high response rates. So we want to use our rare cancer samples, and build a database, genomic database, immune database that we can utilize at a community level. So what's next in our pipeline? We want to be able to analyze more cohorts. Actually, all responders versus control is our next plan. And we want to explore finding more predictors, not only for response survival, but also for immune-related toxicities to this combinatorial approach. So next step, we have plans to use mass spec-based serum proteomics, AI-based radiomics, AI-based pathomics, using the H&E slide TIL assessment, and then, of course, phenomics. So this is data coming from my lab that the serum proteomics has the potential to predict better response and delayed time to immune toxicity. So another work from my lab looking at the radiomics. If you add the clinical variables as well as genomic variables, you can enhance its predictability for response as well as toxicity. So take-home message. Yes, we can accrue rare cancers and make impact, change guidelines, but this still remains high unmet need. So we have DART2 in preparation. And we are learning from multi-omics, translational medicine efforts from DART tissue and blood samples, CMAC and CIDC, SWOG. Team science is in action right now. Stay tuned for making portraits of rare cancer TCGA immune landscape. So for personalization of combinatorial immunotherapy, AI-based comprehensive biomarker discovery and modeling can be applied utilizing multi-layered data points from DART trial. So with all that, I will close my talk, and I want to give thanks to all the collaborators. I want to thank, of course, Dr. Kurdrak and Dr. Patel, my co-leads in running this trial, and the SWOG leadership, as well as the NCI, CTAB, CMAC, and everyone who worked so hard to open and keep this trial alive and active. And most of all, I want to thank the patients who participated in this trial, as well as their caregivers. Thank you.
Wonderful work. And I've heard 2 talks now where we're not talking about the GI center or the breast cancer center. We're talking about biology and trying to address biology rather than the site of origin. That's quite interesting. And as we move to immune therapy for the last part, I think we're going to probably hear more of the same. So do we need to change our entire infrastructure in our cancer centers? Of course, surgery, you still need surgery as a surgeon. I want to say that.
And
But
radiation.
do we need to, once we get past surgery, do we need to go to a RAS clinic or a, MSI clinic or whatever?
So I think there's a room for pan-tumor approach like this,
There's no real answer to this.
but histology, is definitely a big factor. So
I'm sorry, at the microphone. Thank you.
Yes, Sarah Gabriel, Memphis, Tennessee. So first of all, congratulations. Fabulous talk, incredible work. Congratulations.
thank you.
I'm particularly struck by a throwaway line in your last slide. 62% of the patients accrued came from community programs.
Yes.
So how did you figure that out? How did you manage that? This is much closer to the representativeness where 85% of all cancer care is delivered in community healthcare systems, whereas a nearly unnoticeable proportion, right, getting to clinical trials. How did you manage this? And is there something we can all learn from you?
So I guess this is not that different from the NCI match, where you use the existing NCTN and then the community, the clinical trials network, precog, and I think because this had no biomarker requirements, or this was pretty easy try to open, and there was such a hype about immunotherapy, and then you don't have immunogenic tumor, and no pharma is interested in enrolling rare tumors, and we had this trial open just at the right time, and I think a lot of sites were interested and were able to just get it open through the existing NCTN network. So yeah, I'm just happy and grateful that we have that infrastructure ready. And if we work hard to get it open, I think we can not just start, but any other pan-tumor trials can achieve this success.
Of course, like incredible work to get that study in rare tumors, which I think, we need more data on rare tumors. The question is like in cohorts who had good responses, and you mentioned adrenocortical and had a patient with adrenocortical who had very good response. Is there any, this is cohorts of kind of series of phase 2 studies. Is there any way to get like approvals? I don't know. It's very hard to run a big study. Is there any way to get approvals for immunotherapy for these cancers?
Oh, you mean running a separate trial for these
like
entities?
A pathway to get approvals, because, the FDA is not going to approve these or.
Right. I think DART is still a signal finding study or hypothesis generating study. However, if we have enough interest, I did pursue anaplastic thyroid. We had 4 out of 2 patients is living, out 2+ years, one very deep PR. So I did try to pursue having a separate trial open. So I think opening up a separate trial is the pathway for approval at this point, because right now it's actually, the Simon's 2-stage design. This trial was designed to have 16 patients for each cohort, but you know how these trials operate. You open it, and then with the closure date, we're going to close in the next 1 month, and then 30 patients comes in, So that's how we over accrued, but it's amazing that there is such a great interest. And yeah, I hear your enthusiasm. Thank you.
A quick question and a quick answer. Thank you, Rizal.
Yeah, I had a couple of comments. I'm Rizal Kurzrock. I'm the chair of the committee. My hair is blonde now, but old pictures. But I had a couple of comments. First of all, Dr. Young-Chai, that was a fantastic presentation. You've done great work. Regarding the community sites, I think that SWOG is able to open at over 1,000 sites, and that allows patients in the community to get trial close to their home. So I think this is unique to the cooperative group. And the second question, I wanted to answer Dr. Lee Ellis's question about histology. So I'm going to say something that is very controversial, but I'm still going to say it. I've been working in precision genomic medicine for more than a decade now, and I think organ of origin-based cancer is an accident of history, because the light microscope was invented in 1590 and genomics didn't come into play until 2001. And if it had happened in the reverse order, I don't think we would be doing organ of origin. It gives context, but it's not the driver. So there's my controversial statement. Answer.
And I'll say we still need surgeons for the primary tumor as a surgeon. But after that, I can agree with that. Quick comment.
Oh, thank you for the comment. Yeah.
Okay.
Yeah.
Thank you.
Thank you.
That was great. So 2 great talks. I'm thrilled at the speakers and the participation of the audience. Now we have something completely different. After the last meeting, I had asked my friend and colleague Ignacio if he wanted to talk about CMAC, because I heard a lot of people who weren't quite sure how CMAC was working. Ignacio then thought it would be good to include the NCI. So Helen Chen is here, an old friend from the days of angiogenesis. Remember angiogenesis, people? Yeah. So we co-chaired an angiogenesis meeting together in La Jolla. And then Helen wanted to invite virtually Magdalena Thurin from the NCI. So we're going to have 2 15-minute talks, and then we're going to have a panel discussion with our 2 speakers as well as NCI headquarters. So Ignacio, the podium is yours. I'm sorry, Helen, the podium is yours.
Okay, thank you, Lee, for the introduction, and thank you for the opportunity to present. You already heard CMAC. So I'm here to give an overview about the network. So we have the NCI project manager, director, Dr. Magdalena Thuring, Ming Song, and David. So I'm really presenting on their behalf. And as mentioned, Dr. Thuring is actually online. She will be joining us during the panel discussion as well. So I was actually here about 5 years ago. So it's good to be back and to provide some updates and what the renewal CMAC network is going to look for. So why do we need something like CMAC CIDC, such a biomarker infrastructure? So we already heard the biomarker is essential to further development of immunotherapy. However, a lot of work has really limited in the translational potential because a lot of times the trial may have delayed or lack of access to the funding or the very comprehensive assay capabilities are needed for immuno-oncology. And then the individual trials may have limited power given the complexity of the immune biomarkers. And while multiple trial data may be important for combined analysis, there are variability in the assay and also the lack of really a common database to integrate the data for a combined cross-trial analysis. So the NCI had the initiative for the CMAC CIDC with a goal to have a network of laboratory centers in order to provide comprehensive immune profiling and for immunotherapy trials using state-of-art and more importantly, harmonized assay, and also to establish a central database to allow for within and cross-trial analysis. So here with this network, our goal is really to not only enhance the translational study within individual trials, but also to let this work to leverage and contribute to collective power across trials in the networks and organizations. So the CMAC network was launched in 2017 with 4 laboratory centers at MD Anderson, Mount Sinai, Stanford, and Dana-Farber. And each center had its multidisciplinary team in order to carry out the whole spectrum of the biomarker analysis and also comprehensive profiling. And the CIDC is in one center, used to be hosted at Dana-Farber, but now it's moved to the NCI contractor. The funding of the first cycle of CMAC CIDC was actually from the Moonshot Initiative, which is also coming with a 12 industry sponsor to match the funding. The network has just recently been renewed for another 5 years up to 2024, and this time with a NCI U24 funding. So this is a timeline for CMAC CIDC. As you can imagine, this is a really unprecedented biomarker network infrastructure. So it took about 2 years to establish the operational process and also to have the assay harmonized and validated with all the SOPs available to the network. However, we started the selection and prioritization for clinical trial collaborations so that when the infrastructure is ready, the actual assay can take place to generate data after the year 2. So this is really one of the major accomplishments is to establish this organizational infrastructure. As you can see, this is a pretty complicated structure with multiple stakeholders, and within each, it also has multiple parties responsible. Probably the most complicated part is the interactions between the clinical trial team and the CMAC team in order to come up with a biomarker plan, getting the study approved by the CTAB and also at the organizational committees, and then have the biomarker, the biobanks to transfer samples and have the clinical data extracted to the CIDC. So that requires a lot of agreements and SOPs. But now the system is functional in order to realize really the translational studies. So the network has assay capabilities in the 3 tiers. The tier 1 assay is really a basic module applied to most clinical trials. And tier 2 assays are applied to some of the trials. And tier 3 are more novel assays, which are more exploratory. For all the assays to be applied, all the assays actually here have been analytically validated. And for tier 1, there's also, because it's a basic module for all trials, it does require harmonization, which was also completed. And the panel, of course, are evolving in keeping with the field of the immuno-oncology biomarkers. Also, the group had developed a very important specimen collection umbrella protocol, which can be used by the community, actually, not just for CMAC studies, but also really to ensure pre-analytical requirements for immune profiling. To date, there are 37 clinical trial collaborations up to about 2,600 patients. These are the trials mainly from our NCI clinical trial networks, both from the ETCT and early networks to the late cooperative groups. These have expanded from early trials to late randomized studies involving a multitude of therapeutic regimens and clinical settings. Collectively, these studies would address important biomarker objectives, the immune modulatory effect of the IO agents and IO combinations, and look for markers of response and resistance for novel approaches, and was a focus on immune combination therapies. And as I mentioned, to also enhance understanding of rare tumor and which are otherwise understudied. And this is also an opportunity to look at mechanism markers for IRAEs, where we would have the synthetic cohorts across all these trials, and also with some prospective studies for autoimmune IRAEs. Ignacio will be talking about some experience with the SWOG, the collaboration with SWOG. I just want to show one example from the ETCT and early trial. This is a phase 2 study looking at the combination of radiation with immunotherapy doublets PD-1 CTLA-4 in patients with refractory disease. So the radiation has been proposed to potentially enhance immune response, but actually the dose and schedule is uncertain whether it's immune compromising or immune enhancing. So this trial had a randomization for high or low dose radiation. So the trial clinical endpoint actually is an active trial. In the colon cohort, there was no response. In the non-small cell lung cancer trial, there's no difference by adding radiation to the immune backbone. But the trial does provide the opportunity to do translational work on both the baseline and on-treatment biopsies by the CMAC team. And because CMAC was able to work on this trial almost immediately after enrollment completed for these trials, the trial was able to publish its first primary paper with not only the clinical data, but also the intensive biomarker data in lead journals. Some of the key findings from this is that there seems to be a differential immune modulating effect by either low dose or high dose radiation, and also the impact in the tumor cells, tumor microenvironment versus a peripheral seems to be different. This really highlighting the importance of dose and schedule for these combinations. And in the non-small cell lung cell cohort, there are durable responses and seems to be associated with the PD-1 positive and CD8 cells. So this, of course, is still the very limited finding from very few patients, but the system does provide the opportunity for cross-trial analysis for the existing studies. So really highlighting the advantage of such a network approach. So far, there are a lot of trials have been published already or accepted in major journals. Many works are still ongoing. And importantly, for the CIDC database, it had ingested many trial biomarker and clinical data, and as well as providing the assay pipelines to allow for future analysis. So talking about the new renewed cycle, it's now, as mentioned, up to 2028. The goal of the renewed CIDC is CMAC CIDC is to really capitalize on the infrastructure already established and to expand we have the opportunity to expand the collaboration in high priority areas in order to really enhance the scope and process of cross-trial analysis, and ultimately to also develop the capacity to share the data to the public. So this is a scientific themes which is very similar at the high level for the past and future CMAC CIDC. We want to continue to look at mechanisms of the novel agents and combination partners, and more importantly, to have a deeper look at the resistant mechanism beyond just biomarkers for responders and non-responders. The goal is really to understand what are the diverse mechanisms behind the non-responders so that there may be therapeutic strategies that can be hypothesized. So in order to achieve these goals, we are looking for additional trials and to potentially focus to be able to expand on the cross-trial analysis, and just for an example, to have some synthetic cohorts by either the disease setting or the treatment strategies or the mechanism of resistance. We don't really have a strict criteria for what kind of trials, but it can be early to late trials, and with trials in immune resistant settings or neoadjuvant settings. Because there is still a time limit for the funding, all trials do have to have samples available by the end of 2025. So, and ultimately, the larger goal is to really share the data with the public. NCI is currently developing this Cancer Research Data Commons, or CIDC, which will integrate biomarker, clinical, and intervention outcome data to allow the public to have a secondary and larger analysis. Currently, the CIDC functionality is only allowing for within the CMAC network analysis with the trial investigators. But we want to build the capacity so the data can be seamlessly integrated in the CRDC. So in summary, the CMAC CIDC network has established a very unique biomarker resources to enable coordinated translation efforts across trials and organizations. We have come very far with the infrastructure, but of course there are still room for improvement to optimize the process and the efficiency. The renewed network will expand trial collaborations with a focus on high priority areas. So this will further enhance the IO trials, both individually and collectively. And we would like to really contribute to the larger collective efforts with other organizations, because what CMAC can do is still limited, but we're looking to the larger research community in order to really accelerate the development of biomarkers that are not just published in the paper, but can be clinically impactful. So lastly, I just want to show this slide to thank the very large group behind all this effort with the CMAC centers and CIDC, the PIs, and the very dedicated staff working very hard on these trials, some of which you just heard. And also to thank the trial teams, because this is a collaborative efforts. All the process involves a very close interactions between the CMAC and the trial teams, and also my colleagues and at CTEP and the Foundation NIH. So, thank you.
So we will not have questions at this point because we will have a panel discussion. So I'll introduce Ignacio Westuba, my colleague and friend from MD Anderson Cancer Center, to provide the pathology and hands-on perspective, and then we'll have a panel discussion.
Thank you very much, Dr. Ellis, for the invitation, and I thank the previous speaker because they're going to make my job easier. I'm going to share my experience on the work that we have done with my colleagues at MD Anderson CIMAC with the SWOG organization. You saw two trials actually that Dr. Chen presented are part of the SWOG in CIMAC. Actually, we're very fortunate to actually take them both in our group to lead them. So we're all looking for biomarkers in immunotherapy, right? And there is a lot of research going on in this field, but we don't have many. And that's why I think efforts like CIMAC are important. And we're trying to get biomarkers to actually predict the effect of the efficacy of immunotherapy approach in our patients and actually maybe spare some of them from adverse events that are resulting from this. So the field of cancer translational research that I've been working over the last 30 years have expanded now from classic pathology to molecular to immunology and many other approaches that I was not familiar with. We have to learn and optimize in laboratory, and we have not to look at just the tumor. We have to look at the patient, because in the immune response there are many host factors that are very important on the immune system. This was already shown, the CIMAC network. I'm not going to go in detail on that. We already have shown the TR assay approach, and I think that TR3 are becoming more important now because one of the activities in the new CIMAC or version 2.0 is going to bring more innovative assays to the network, and we already have Dr. Chen talk about the validation and harmonization process of multiple assays, and our CIMAC participating actually in all of them. Our team, you can see here, is large, and some of the people are listed. Dr. Haymaker, who is in the room, is one of the co-PI with Dr. Gia Thaladrash. And we have very fortunate to have Dr. Jack Lee as a biostatistician, because statisticians are, very important in all this work. And I would like to highlight at least two CIMAC that help us in the two trials. I'm going to show some highlight of the result today. Dr. Maker from Stanford CIMAC and Dr. Nyatik from Mount Sinai CIMAC. And I would like to thank also the clinical investigator from the two trials. I'm going to show some data on the DART trial that Dr. Chai already presented some, and on the landmark S1400I cohort that we were able to work. So, and this work actually was not easy to start, and Dr. Chen actually presented this in a timeline, and actually every single activity there took a lot of effort, particularly we have to go through the COVID pandemic. And the challenges I would like to list here basically is that in all this process, we have multiple level of approvals, and we are adding actually for the CIMAC network additional approval, a lot of MTAs, you can see. So the availability of the specimen in some of the already ongoing or finished clinical trial are not always the best for immune profiling or even innovative approaches. There are some other data that our colleagues in the clinical trial have committed or acquired, how we can integrate them. The availability of the clinical data for our CIMAC laboratory to do quick analysis and look for biological correlation is not always easy, because we understand the clinical trial needs to be finished, the endpoint needs to be analyzed, and the publication needs to be done. The biomarker publication strategy actually is something that we have a serious guidelines in our CIMAC network, but require a conversation between the CIMAC team and the clinical PI, so we all, acknowledge the people who have done work. And it's not, the resources are always limited, and the bandwidth is not as big as we wanted. And this is some of the challenges. Probably you can add more. We can add more if we start talking about that. But I see challenges, opportunities. So What is great about CIMAC have been shown is comprehensive standardized molecular immunology profiling assays available. I think that we integrate this with the CIDC site with the clinical data. There's a very, very good collaborative environment between the CIMAC sites. And that has been growing over the years. I really appreciate that. And there is more innovative approaches coming to the CIMAC 2.0 more than the CIMAC 1.0. And the collaborative approach in our case for this context of this presentation with the SWOG investigator, clinical statistician, translational researchers has been outstanding, but we can do more. And how we can do better, I think that talking more about how we actually face future clinical trials in our CIMAC network or on this MD Anderson CIMAC represented today. So I'm going to talk briefly, and this is going to go very fast in the first clinical trial because that was presented by Dr. Chai, and also I know this was discussed extensively by Dr. Kara Heimaker, our immunologist in the team, who has a lot of experience in oncology and immunology, and she presented today in the ET-RT committee session. And this is about 2 cohorts that have been already mentioned on the neuroendocrine tumors. And here I'm showing this sample availability for analysis because it's very important to keep this in mind. These are retrospective collected specimens that have been already examined for other abnormalities, including the match trial analysis. And here you can see that if you list all the potentially assays that we can do in tissue and blood, there are a number of cases actually that don't have all the parameters that you like. So that's one of the issues. And in this case, it's 46 patients. So it's the limit, the analysis is limited, and we consider these studies exploratory. We already learned that PD-L1 expression immunohistochemistry in this neuroendocrine cohort or the cohorts of the DART trial were not predicted, but B cells infiltration and lympho aggregates were important. And I would like to actually talk a little bit more because it's also the theme of what I'm going to present for the lung cancer trial, the LungMap S1400I, because we are doing a lot of spatial analysis. And it's not just the cells that are infiltrated or not, a tumor, it's the location of the cells. And now with the use of digital pathology, machine learning, and spatial analysis, we can actually look at this in detail. And this is a summary that show for this cohort of neuroendocrine tumors in the DART trials is that if the immune cell cluster, this lympho aggregate of B cells, they're found closer in the tumor cells related to the tumor cell in the responder and more in the stroma in the non-responder. So the location and the proximity of these cells to the malignant cells is very important. And here's a summary of what we have learned in the DART trial. I'm not going to go in detail, but I would like also you to keep in mind that not just the number of cells, the type of cells, where the cells are in the tumor is an element that we need to keep in mind in any of our analysis. So the second trial is this phase 3 S1400I, a cohort of the LungMap consortium, in which you have an anti-PD-1 and anti-CTLA-4 arm to test the hypothesis if this combination could be more efficacious in the overall survival of patients compared to a single anti-PD-1 or nivolumab. So this trial enrolled 252 patients. The endpoint was not achieved, so the combination of the two agents didn't show better overall survival than the single agent that published in 2021. So we're able to work, and this is the work done by Dr. Edwin Parra, Jay Zhang, and again, Dr. Heimaker in our team, and in collaboration with our colleagues from Mount Sinai, CIMAC, we performed PD-L1 immunohistochemistry, multiple immunofluorescence using 2 panels, analyzing immune cells in the tissue, whole exome sequencing, nanochain RNA instead of RNA sequencing, because the quality and the amount of material that we could collect from these FFPE tumors, and a panel of about 100 cytokines, immune cell activating factors in the blood that was performed at Mount Sinai. So I'm going to highlight some of the findings, and mostly because I'm a pathologist, I have a bias. I'm going to focus on the immune infiltration of in the tumor and in the data of 17 cell phenotype that we define into panel of 7 or 8 marker each in the multiplex immunofluorescence. The issue is that out of the 252 patients that you actually I showed, we have only 82 tumors that we actually run at baseline our immunofluorescent panel, dividing 35 from the 2 drugs arm and 47 from the single agent. And there is a series of markers, immune cells that are associated with better outcome, particularly in the NIVO trial, in this case, progression-free survival and overall survival, and are cells that have memory and memory regulatory features. And in the combination arm, we found that a couple of activated T cells or T cells that experience antigen actually were associated also with a higher expression of baseline with better progression-free survival. But what's intriguing to us is this higher presence of regulatory T cells that associated with worse overall survival in the combination arm. And this is something that we would like to follow up. And there is a clinical trial run with the same drugs in Italy, Dr. Federico Capuzzo, and we are going to do some collaboration with him to follow up in this finding. And also talking to a colleague from MD Anderson working in this area, there are some in vivo models that actually could help us to understand this better. And this finding, actually, you can see here the couple of measures that we have. But in the theme of spatial analysis, what we did in this trial is to study the distance of immune cells to malignant cells, and to see if we can actually understand better the pattern of infiltration of immune cells and correlate with response outcome of the patient. And we found that for all cohort and for the combination drug arm, not for the single agent, that certain phenotype of immune cell, when they were closer to the malignant cell, actually will improve progression-free survival and overall survival. And these were when cytotoxic T cells or activated cytotoxic T cells were closer to malignant cells. And some of them even expressing PD-L1. So that makes sense, right? So, but it's important to keep an eye on all this interaction of the immune cell with the malignant cell or immune cell among each other to have a better, actually, perspective of how the immune system works with the intervention that we have. Then the group, and I'm highlighting here Dr. Kumar and Dr. Zhang, that have bioinformatics, expertise with the help of the clinical investigator. What we do look at is the effect of this spatial infiltration of immune cells in a group of patients that have best or worst response in the trial. And these are all cohort. And we divide the patient and we call exceptional responders. And when they have no sign of progression at 18 months, and they were alive at 24. So those were called exceptional responder. Don't blame me about the name, because I already have heard some criticism about that name. And then we selected, that were 11 patients, and we selected a group of about 30 patients that we call early progressors, that were alive after 1 month, and they have progression in the first 6 months. And then we look at actually the infiltration of these cells that we found that were associated with the outcome of the patient. And we found that actually by doing nearest neighbor analysis, that when we have an infiltrative pattern and these cells are integrated or inside of the nest of malignant cells, actually associate with the exceptional responders. And when these cells are not clustering with the malignant cells, are far away, and we kind of excluded, we associated with the early progression. That's the simplest way to explain this. We also found that genomic abnormalities like copy number burden established by whole exome sequencing associated with the cold environment by RNA analysis or multiplex immunofluorescence, but they didn't correlate with any of the outcome. And I'm not going to go in detail on the analysis of the 92 proteins by in the serum, in which we found that immune activation and primers are expressed in responder, and marker of macrophage-derived hyperinflammation and tumor-derived markers are expressed in non-responder. So this is one of the caveats of the study. Not many samples, actually we have multiple analyses, and the one that we had the most was with multiplex immunofluorescence and OLIC, and we tried to develop a predictor that actually is not very good because we don't have many samples. So we learned a lot from this study, already explained that, I'm running out of time, and I would like to just go to the conclusion slide, saying that exploratory biomarkers in the S1400I lung map show interesting exploratory and mechanism of response in this combination of therapy in the DART trial, where actually you already where that was explained to you. And importantly, we have demonstrated that CIMAC network and SWOG can work successfully together. And I think that we can have more work in the future. And the MD Anderson, CIMAC, and the entire network have been renewed for 5 more years, so we have more opportunity. And thank you very much for your attention. 1 minute over. That's a lot of work.
All I could say is, wow, that's a lot of work, and there's a lot of interpretation to do. Let me see if I can unmute Magdalena.
Actually, I can unmute myself. Thank you.
So Magdalena, we can hear you, so that's great.
Yeah, thank you.
We have our 2 speakers up on the panel, and I am looking for David.
I have a question for Ignacio, and it's about the last part of your presentation, identifying responders and non-responders by the time. And my question is, there is a phenomena called hyperprogressive disease from checkpoint immunotherapy that it's very controversial, but if you believe in it, there is actually activation, or I should say there's progressive disease that is due to the checkpoint inhibitor. And there's been work looking at tumor-associated macrophages as maybe mediating that. Do you think this sort of analysis, because the Gustave Roussy has hundreds of these specimens that they've classified as hyperprogressive disease, and there's no biomarker. We did an analysis of 800 patients looking at all sorts of things like EGFR and TMB and so forth. Nobody has really centered on anything. Do you think maybe this analysis could identify something where you could prospectively exclude patients from having actually progression as a result of the checkpoint inhibitor?
I think that that's a very interesting thought. I know that is a controversial issue, but I think there are many series of people are getting together and that could be explored by this type of analysis. I think that there is an opportunity to bring actually higher multiplex platforms to this analysis. We did this multiplex immunofluorescence of 8-9 markers each panel, but now we are all running panels of 40, 60 immune cells using other technologies, and you have even the opportunity to do spatial transcriptomic. I think that those analyses actually can help to understand what is the immune status of patient at baseline, and that could predict that type of behavior of the tumor. And I'm happy to actually even explore that. And you need to correct me if I'm wrong, but this is not just for lung cancer, right? For other solid tumors. So it's an opportunity to look at there are similar mechanisms that happen across different tumor types. And actually, that's one of the value of a network like CIMAC at some point, and we can do even, a clinical trial analysis of similar mechanisms of using the modality of that we're using.
Thank you. Maggie Senthil from UCI. It's just mind-blowing as to what you have shown us at this point. One, I have two questions, if I'm allowed. One is a logistics question. So as new investigators embark on studying immunotherapies, whether it is in the neoadjuvant setting or metastatic setting, when and how should they engage with CMAC-CIDC? And also, do you have a guidance document that allows them to plan their correlative science and ensure that the specimens are being collected with appropriate quality for future, whether it is multiplex IF or site-off? What guidance do you currently have to provide to them?
Magdalena, do you want to answer that question?
Yeah, and I can answer. So hello, I'm Magdalena Turin from the Cancer Diagnosis Program at NCI, and we're running CMAC-CIDC network together with Helen and other investigators. Well, this was mentioned really very briefly that CMAC-CIDC project has limitations based on the funding. Now we renewed the network for another 5 years, but this is via grant mechanism. So we had to go through, develop the concept and present it to all kind of upper management in NCI to receive the funds. So we can only project work for next 5 years. Therefore, we basically have to accept to CMAC network trials, which are basically completed and have specimens collected. And Helen actually mentioned the deadline for us is 2025. And of course, we would be very much interested in new study design, new agents, and so on. And we are looking at LOIs coming to CTAC for review with new trials, new design and new biomarkers actually, but we cannot accept the trials which are still in development. So that's our problem. So in a way, we are not open for young or established investigators to work with us, as long as the trials are not basically completed or will not be completed by 2025.
I just want to add, so there's an issue limitation of the current CMAC. However, as you can see, you know who to go to, who had the established capability. So what's lacking is potentially just funding. In order to prepare your trial for potential immune profiling assays by anybody, as I mentioned, there is a specimen collection protocol for for tissue and for blood. And this is actually went through extensive discussion within CMAC what these procedures should be. So that's available in our website. So this is something you should do upfront so that when the opportunity and funding comes, you have the right specimen for this.
Yeah, in addition, actually, CMAC develops resources for the community. As Ignacio and Helen mentioned, all assays, including tier 3 assays, which are these novel assays, are stringently analytically validated, and the protocols of the data for analytical validations are also available. So for to use perhaps other resources, these protocols can be useful as well.
The other thing I would like to mention is that, what people working in CMAC, we have developed an special expertise to work with samples and clinical trials coming from the networks, right? So that actually is something that can be shared. We cannot put in the report of the CMAC grant, but actually can be shared. And I'm really happy to, our institution is SWOG institution, happy to share an advice on what would be the best way to collect and process specimen for the future. CMAC or not CMAC, but at least help on the translational research.
No, that's fantastic. Dr. Ellis, if I may, just one scientific question.
Real quick.
Yes. So you touched on translation of this work as a clinically actionable biomarker, right? How are you advancing the science? I mean, it is beautiful to see the multiplex IF images, absolutely gorgeous, with responders. But how would this translate into a clinician's actionable tool? And how are you thinking about advancing that eventually to make that actionable?
Okay, so the data have to come not from exploratory analysis. It has to come from prospective collected specimens. And then even if they come for higher multiplex platforms, like, several that are available, you need to, for practice, unfortunately, you need to limit it to a smaller number of markers that you can run in assay that people in the CLIA labs are comfortable with, right? So pathologies in the diagnosis are in the diagnostic field are comfortable with immunohistochemistry. We're getting more and more comfortable with multiplex immunofluorescence. And I think that the platform that exists now are things that can be validated in the CLIA labs and could be used in the future for potential biomarkers. And if we add that pathology is going to digital pathology and computational pathology in the future, I think that they're going to get together. So multiplex analysis and digital pathology in the future, in my opinion, are going to be part initially of clinical trials and future biomarker, but that needs to be narrowed to a more limited number of variables.
Certainly a molecular profiling tour de force, so congratulations. Dr. Westuber, I'm just a little curious on why you decided on a 700-odd targeted panel with the nanostring assay rather than going full transcriptome. An argument could be made is that maybe you're not discovery-based now.
Yeah.
And the library requirements and stuff is not really that different.
Yeah, I agree. And the reason I explained very briefly, and I was rushing probably at that part of the presentation, is that in our experience in the initial trials in which we had to work with samples that were used and kind of exhausted for previous biomarker analysis, like the S1400I, the quality and the amount of RNA that we could extract from the residual tumor tissue was very minimal. So even if we are aiming for RNA-seq as one of the tier 1 assays, we put as an alternative a limited panel of 770 gene by nanotring, because we know that we can run that with 200 nanogram or even less of RNA. For RNA-seq from FFPE, you need at least 400, 500 nanogram, if I remember correctly. So that's the reason. It's the only thing that actually worked in those specimens. But in the future, we should have more RNA-seq data and hopefully some in spatial transcriptome and data.
Yeah, so Ignacio mentioned the list of challenges, and one of the challenges is the quality of specimens. As I said, some of the trials that we're working with have years-old collections of samples, and unfortunately, this is one of the sort of technical challenge for especially RNA-based assay or RNA sequencing.
If I'm allowed, I think that at the beginning of the network, we spent, and the timeline showed, a significant amount of time actually setting this network up. And then we wanted to do work with actually real samples, and we had this hybrid approach. Let's get whatever we can get, that a trial that have good collection, a good question that we can analyze. But we couldn't run all the tier 1 assays. For example, in S1400I, we didn't have peripheral mononuclear blood cells. We couldn't run cyto. So we have to compromise at the beginning of the network, but the newer trials are coming with larger number of specimens that's suitable for the entire, tier 1 suite of assays, and even some more innovative from the tier 2 and 3.
Yeah, we're also adapting, for example, single cell sequencing assays for FFPE samples, including single nuclei, and actually single cell sequencing. So we hope that we'll be able to adapt this new assays even to old samples.
So the network and the collaboration is beautiful, great. The support from the NCI is wonderful, even just here on this panel. I want to put my surgeon's cap on and say, how do we predict who's going to respond to therapy or not?
That will be the goal of Holy Grail.
So how do we get to that point? And do we have to prospective trials? Do we need core biopsies? Do we need excisional biopsies of tumors? Because Ignacio, you talked about the spatial relationship between the immune infiltrating cells, if I can use that broad term, and the tumor cells.
I thought they were friends, and you're asking a difficult question. So, okay, so I mean, and in my experience working at MD Anderson, one thing that I've learned is that with immune response, and also we learned with the genomic, targeted therapy analysis, that longitudinal follow-up of these events is very important, right? And I strongly believe that we're going to learn more. We go beyond baseline analysis of tumor tissues to understand the playbook of immune system in oncology and intervention. So if we have more in the academic groups trial with ongoing collection of tissue, I think they're going to learn more. And then we can go back to the baseline, which we're going to collect mostly in the network trials, right? And then look for the sign that we don't see clearly at the beginning. I've seen in several types of tumor that are so-called cold tumor. And then after a couple of cycles of immune checkpoint inhibitor, some of them are not cold anymore. So how we capture those? Because those are the ones that surprise us when we look at biomarker at the beginning and they don't have what we expect, but they have some response to stable disease. So I think that that's one thing that we could do. The other thing is that, as happened in the genomic field, now we use a lot of liquid biopsy because we know what we're looking for in a diluted environment, like blood, right? Genomic analysis. So I think that in the future, hopefully, in immuno-oncology, if we learn what happened in tumor, we can try to identify those markers diluted in the blood, in the patients, right? That now is so confusing, right? There have many other things going on that is very difficult to sort out what is an event associated specifically to the response of the lack of response in a biomarker in the blood, serum biomarkers or immune cells. So a couple of things that I think are going to help. And the third, with my pathology hat, is the revolution of computational pathology. They're going to help us to better understand the interplay of the malignant cells and the immune cells in the tissue with basic or very more complex molecular or approaches.
Mm-hmm. Yeah, so I think this is a very difficult question for immunotherapy because the nature of the mechanism of action. It's not like the target agent, you have a drug target. And this is really, you don't have the mutation in the immune system itself that you can define. So I'm not sure if we will have a truly precision marker, very precise marker. We can probably have a categorical marker that certain immune microenvironment may respond to certain kind of immune therapy or combinations. But even that will require a huge data set to do the analysis simply because of the complexity. And I hope that we will generate a lot of data so the AI and machine learning can help us eventually. But that does require a lot of data.
Yeah. And as Helen mentioned at the end, the ultimate goal for the network is to create a database, and the database will be used by outside investigators for secondary analysis. But also, we think that we will not end up with a single biomarker which will be predictive of response. Therefore, we also think about developing new bioinformatic approaches to create signatures which will sort of integrate tumor and host and tumor microenvironment. This is really extremely complex system, but with the help of computational approaches, AI and so on, we can make progress.
So we're running a bit over. We do have one more question, and then I think we'll end. We have two more questions. I'm sorry.
Thank you, Lee. I'll make this brief. So fantastic presentations. Thank you. I feel like I have to go back to medical school again. Probably should anyway. But I had seen a presentation looking at the tumor microenvironment using an animal model with video microscopy in real time, looking at labeled infiltrating lymphocytes. And I didn't know if that was several years ago, if there's a potential to combine animal models looking at the tumor microenvironment to try to get to the 3D modeling you're talking about, or is this so varied that trying to use animal models will not be helpful for us? I mean, we've learned so much in years past by very crude animal model experiments with chemotherapy. There's the potential to use video microscopy, learning about the immune response in animal models and how effects on inhibiting PD-L1 and so forth may give us some of the biomarkers you're looking for.
So I'm not the right person to answer that, but I'm here. So I'll give it a try based on my experience working with multiple collaborators at MD Anderson. I think that, again, and probably my bias, I think that we need to understand more of what happened in human tumors by deploying all technologies possible in tumor specimen on longitudinal basis. And then from there, to understand the mechanism of what we see, I think that animal models and can help. And I have just an example mentioned today that about this T regulatory cells associated poor outcome when you have a combination of drug that I showed. Actually, a colleague was talking to me about the model that they're looking in the mouse that they found the same, right? And similar findings. So we will need to talk and check notes. But I think that is potentially a way where I see the animal model can help. But that's my
quick question, then we need to wrap it up because there's other sessions that are ongoing this evening.
Great discussions. And I see the path for the near future. That is a dream coming true, but I did not hear very much about the influence or how to address microenvironment of the tumors. Do you have any insights about that?
You mean therapeutic interventions or the biomarkers?
Both.
Yeah.
Well, we know a little bit about TIL,
yes.
but we don't know much besides that.
Yeah, so I think one way we are approaching this is, as mentioned, a lot of, majority of the patients do not respond to immunotherapy. And there's a lot to learn what is in the microenvironment or the intrinsic tumor markers that's responsible for that. And you can imagine it could be very diverse and multiple. And so that's one thing. And the other is to study, we have thousands of clinical trials for combinations with different mechanisms to change the microenvironment. And by doing really the intensive biomarker assays to look at how they really modulate the microenvironment and the immune cells, maybe we can start to learn that which kind of mechanism of resistance and microenvironment features may be addressed by certain drugs. So that, yeah.
Very briefly, I think that one of the limitations in metastatic advanced disease is the amount of tumor tissue that we can analyze to have a full perspective of the heterogeneity of the microenvironment, which are our best stroma immune cells, right? Or malignant cells, that's it. But there is an opportunity in surgical resected tumors in the context of neoadjuvant immunotherapy, in which pathologists actually, they have the entire tumor and adjacent a non-malignant tissue that it actually have some immune response. And then people are talking about tertiary lymphoid structure that happen in adjacent non-malignant lung tissue, for example, in the context of non-small cell lung cancer, that may play a role. Or also the lymph nodes that are resected at the regional, lymph nodes that are important in all the immune response. So I think that there is opportunity to learn more on the context of the earlier disease that are treated with immunotherapy and resected.
But we have several essays, the spatial transcriptomic, spatial imaging, which address morphological combination and functional and phenotypical features. So we definitely think about microenvironment.
So more work to be done, no doubt. This was a great session. I know people are leaving the room to go to their next session or to go to dinner. I want to thank Magdalena for calling in and participating, and Helen and Ignacio. What a fascinating session this was. And like I said, more to come in the future. So thank everybody who participated. Thank you for those who were able to stay, and we'll follow up on this. Thank you all.
Thank you. Thanks very much. Bye-bye.
Thank you,